discussion today will do some betterment for the society. I would request the admin team to please allow me to share the screen. Right. Okay, I guess my screen is visible here.
Yes, it's visible. Please put it on slideshow mode.
Right, we'll do that.
Thank you, Vijaya. All the best.
Thank you, ma'am. So the topic for today's discussion, which I'm having, is the modified or the modification of carbohydrates for better glycemic control. Now, while we talk about carbohydrate, like we know that this particular macromolecule is very much needed for every human body. Without carbohydrates, neither our brain nor any cell of the body will be able to perform any function. So carbohydrate is a must in our diet form. But on the same side, it is even true that a free release of carbohydrate can be deleterious. It is being noted that if there is a free infusion of glucose in blood or fructose in blood, then that can lead to multiple diseases, starting from fatty liver, diabetes, obesity, and a lot more. So we must be aware that what are the right ways of consuming carbohydrate? What are the techniques that we can do at our end to utilize carbohydrate in the correct manner? So while we discuss about carbohydrate, the most common form of carbohydrate remains starch. Starch is obtained in two major variants, that is amylose and amylopectin. The structure here depicts that amylose is a straight chain, whereas amylopectin is branched. Amylose with a simple structure becomes more stable, and the release of glucose from amylose is comparatively slow than for amylopectin. So if we are choosing a starch molecule, we must be aware that what is the ratio of amylose and what is the ratio of amylopectin. Because more the amylose, less will be the glucose infusion. There is a common technique while we cook the starch, which is called as gelatinization. This is a phenomenon which happens whenever any starch molecule is being heated in presence of water. Whenever the starch absorbs the water, it swells, it ruptures, and it releases tiny amylose molecules, which makes it as a gel. This gelatinization makes sure that while digestion, there is a slow release of glucose. The same gelatinized molecule can be converted into the resistant form of starch. Now, there are multiple categories of resistant starch. This phenomenon will lead into conversion of a third category resistant starch. Now, this group of resistant starch, they are very specific as because they are not completely fibrous. So they will degrade, they will release glucose, but on a slow mode. And then they also help the microbes to grow, proliferate, and release the very needed short chain fatty acids. The third grade starch resistant form, which I'm talking about, it will happen only when after gelatinization, the paste or the gel is being cooled. Now, this technique is termed as retrogradation. Now, again, we have to be aware that gelatinization and retrogradation is mostly done with the amylose molecules as because of the linear pattern of structure. So while we cool any form of gel, it will undergo a stage which is called as retrogradation. Now, this retrogradation makes sure that there are hydrogen bond formation, which make the molecule very dense and there is a slow release. But this phenomenon fails to happen when the amylopectin concentrations remain high. Temperature determines the glucose release. The graph here depicts that how, with temperature, the gelatinization tendency or the viscosity of the starch is increasing. More the viscous the starch form is, it becomes more difficult to break it and easily release the glucose. In French, there's a saying called al dente, which means that while we consume any form of carbohydrate, it should be such that we can chew it and we can bite it. It shows that this pattern of gelatinization makes the starch or carbohydrate such that there is no easy diffusion of glucose. So this is the right temperature that we must maintain. This temperature ranges around 75 degrees to 90 degrees Celsius. If we want to convert the same gelatinized starch into a more viscous or a more resistant form of starch, then we have to lower the temperature. And this is a common practice nowadays, which says that once the rice is cooked, which is of course a gel, then it can be cooled. With the drop of temperature, the viscosity of the amylose will rise and it will turn into a resistant starch. There is another common technique for cooking starch, that is dextrinization. It is commonly known as roasting. Whenever we put the starch directly on flame or we cook it for more than 90 degrees Celsius, then after gelatinization, a new technique will happen which will distort the starch molecule. And that is when dextrinization takes place. As a result of dextrinization, dextrin molecules are formed. Now, dextrins, they are very similar to maltodextrin, or we can say glucose modifier or glucose oligosaccharides. They are small units, as a result of which there will be a very easy release and rapid increase of blood sugar levels. We can note in these two images that how roti and bread, while they are being roasted, they show a change of color. This change of color shows that there is dextrin formation which has taken place. So in comparison to dextrinization and gelatinization, we experience that dextrinization will definitely allow an easy release of glucose in our bloodstream while we consume it. It's not that we always have to look for resistant form of starch. There can be other ways by which we can lower down the glucose release. Like one such example will be cooking white rice with barley. Now we know that barley is highly viscous, though it has its own benefits, but then it's very easy to consume. But in that case, if we prepare a combination of 30:70, that is 30% of barley and 70% of white rice, if they are cooked together, then the fiber which is present in the barley, that is arabinoxylin, it makes sure that the ghrelin levels remain right, which will make a feeling of satiety and it will suppress the hunger. Beta-glucan molecule, which are released when barley is cooked, will bind to the starch of white rice and they make sure that the glucose spike does not happen after the meal. Phenols or polyphenols, which we are aware that they mostly interact with the micronutrient absorption and they lower down multiple vitamins and minerals absorptions in our gut lining. That is very true. But the same polyphenolic vegetables, if they are cooked together with the carbohydrates, then the long chains of polyphenol pair up with the long chains of carbohydrate. And ultimately, we end up getting huge molecular structures. These molecular structures, they are not easily digested in our gut lining. So the carbohydrate automatically start acting like a resistant starch. The good thing is that whenever polyphenols are consumed with carbohydrate, carbohydrate does not tend to easily release polyphenol, as a result of which polyphenols don't get an option to interact with the micronutrient absorption. So like this, we can ensure that micronutrient absorption is right while the meal is taken, and simultaneously the carbohydrate surge or the glucose surge does not happen in our body. There are few more modifications techniques that are mostly used while designing supplements, and they act as non-glucose sources. The very common one is fiber. Like we know, most of the fibers that we generally tend to consume, they are modified forms of carbohydrates. One such fiber is the fibrosol. Maltodextrin, which is regularly mostly found in many of the supplements, is a good source of glucose. As because maltodextrin, they have alpha-1,4 linkage. Now, like we know that the amylase enzyme which we have in our gut lining, they are capable of breaking alpha-1,4 linkages, and as a result, maltodextrin tend to release glucose. But in case we convert the maltodextrin chemically into new forms where the alpha-1,4 linkages are substituted with alpha-1,3 and 1,2, the amylase enzyme in our gut lining fail to break it. So they start acting as fiber. Like that, the fiber will make sure that there is no glucose surge directly and there will be a glycemic control. There is another common modified form of carbohydrate, that is olestra. Olestra is not a glucose source. Olestra is neither fiber. Olestra is basically a modified sucrose molecule which is used as a fat substitute. So olestra is a combination of carb with fatty acid chain. So it starts acting like a fat. So we can prefer olestra while preparing any snack for an obese or a diabetic patient where we don't want any dietary fat to get inserted into the body of the patient. Now, all esters generally don't get digested or absorbed in our gut lining as because of its complex structure, but then it can be used as a fat source for preparing multiple snacks. Modifications can also be done in terms of diet for glycemic control, like we know GLP, that is glucagon-like peptide, which is a common incretin molecule, which is naturally synthesized by our body. Now, this GLP, it is getting synthesized from our gut lining, of course, but it mostly happens in response to the food that we take. It is believed that when we take carbohydrate-rich food, GLP is triggered, GLP is released, and this GLP will ensure that the pancreatic cells releases insulin. Now, the problem is that while we are dealing with any diabetic patient, so definitely there will be carb counting, there will be lowering of carbohydrate. Now, in that scenario, automatically the GLP secretion is reduced. We have to keep in mind that it's not only carbohydrate which promote GLP, other than that, lipids also play a very vital role. So if we keep that in mind, that while designing the diet pattern, if some good amount of fat is added, then that will make sure that GLP release is there, which will trigger the pancreatic cells and maintain the glucose homeostasis. Diabetic patients are also very much prone to gluconeogenesis. So like we know that glucose, while it is in dietary sources, we can restrict it, we can control it, but the glucose that is being produced endogenously within our body, particularly by liver, that is very difficult to regulate. So we have to understand that this gluconeogenic techniques or this gluconeogenic process, when it happens and how it happens. A diabetic patient is mostly in a state of starvation. Why I'm saying so is because during diabetes, like we know, the glucose failed to enter into the cell. As a result, the body can easily go into the stage of gluconeogenesis. It is mostly observed that gluconeogenesis happens before the breakfast hour or in the early morning hour. The same spike can also be seen before the major meal, like before lunch or dinner. So if we want to reduce this sudden rise in the glucose level, then definitely we have to understand that there should be certain dietary source which will allow a slow drop in glucose level. And that can, of course, be done by adding fiber sources and protein. Small snacks which are given at an interval of 3 to 4 hours with good amount of protein and fiber will definitely make sure that the glucose levels don't drop a lot, as a result of which the gluconeogenic pathway will be reduced. So by modifying the diet pattern and the time schedule, we can definitely check the amount of gluconeogenesis which is taking place. There is another common method, that is sequencing the food or determining the order by which we consume the food. Mostly, carbohydrates are consumed first, followed by the vegetables and then protein. Now, while our body is in this hunger response, then the enzymes are the beginning. So at that point, there is an easy absorption, diffusion, and which will result in a higher amount of free glucose. If there is a reversal where the fiber-rich or the micromineral-rich, micronutrient-rich vegetables are given at the beginning, then it will bring a feeling of satiety, and it will also ensure that the micronutrient absorbance is correct. Followed by which, if protein is ingested, then we know that protein makes sure that there is good hormonal response, that is the GI hormone, where cholecystokinin and incretin, GLP, all are going to get synthesized quickly. This will help in easy digestion of carbohydrate when ingested. So just by reversing the order, we are making the body preferable to lower down the digestion, and better insulin regulations are done. As a result, the free glucose release is reduced. There can be few dietary patterns which can run parallel to drug mechanics. Like we know that drugs have their own mechanisms of regulating the blood glucose. Just previously, Shilpa ma'am has wonderfully explained that how different drugs have their own regulatory pathway. Diet can also do the same. I'm not saying that it can compete the drug, but of course, yes, we can follow the same path if we choose the right thing at the right point. I will explain this with an example of a common medication, which is metformin. Now, like we know, metformin typically in liver will lower down gluconeogenesis. It also lower down the fatty acid synthesis. Now, just now we have discussed that how right protein and fiber at right duration can make sure that the gluconeogenic suppressions can be done. Simultaneously, if there is good fiber content, then the fatty acid absorption, the dietary fat is again getting reduced. For our skeletal muscles, the same metformin help to expose more and more GLUT4, that is glucose transporter 4. Glucose transporter 4 makes sure that there is right amount of glucose infusion happening in the cells. Now, with a simple lifestyle change where we can add some amount of aerobic exercise, some amount of high intensity exercise, our muscle cells will be modulated. There will be gene expressions which will trigger more amount of GLUT formations on the cell surface. So naturally, the body will allocate and translocate more and more GLUT if we include the aerobic and high intensity exercise in our regime. So that is how together the lifestyle and the diet can bring change. These are some of my references which I have used.